and good morning. It's my pleasure to welcome you here to this Doce webinar, Beyond Plastic, Pollution in the Deep Sea. My name is Lucy Woodall. I'm Associate Professor at the University of Oxford, and I am a co-lead of the Doce Pollution Working Group, along with Nilia Mestra and Shamik Desputa, who are on this call today and who are also going to be talking to you. But as a way of introduction, let me introduce you to Doce, which is the Deep Ocean Stewardship Initiative. Doce is a scientific network that operates across disciplines and borders to advise on the sustainable use and management of the deep ocean. Doce's got a lot of different working groups and many that address really different topics across the deep sea, and these include things like fisheries, deep sea mining, this working group, which is pollution, climate change, and of course many others. You can find out more about Doce on the website that Hannah's going to put into the chat. So to introduce this working group, which is the Doce Pollution and Debris Working Group, it's actually the newest working group to Doce, and it currently has 78 members from across 29 different countries. Our goal with the working group is really to improve public understanding and promote research into the patterns, the impact, and the relative risks of all different types of pollution, whether these are chemical or physical, and of course, the more well-known versions such as plastic. We also advocate for the consideration of the deep sea into policy development and conservation initiatives, as this is a place that is often out of sight and out of mind for policymakers. And we also provide expert opinion on pollution and debris through written responses, through workshops and engagement with environmental, marine, and pollution and debris focused groups. So it's my pleasure to welcome you all here today. So this event is called Beyond Plastic, Pollution in the Deep Sea. And we have particularly called it Beyond Plastic because, as we said, plastic pollution in the ocean has become a really important topic in the media in the last few years. However, pollution in our deep seas, or in fact our entire ocean, isn't limited to plastic. And there's a lot of other pollutants, things like chemicals and persistent organic pollutants that we can't necessarily see, but are equally as important and potentially hazardous. So our panel today consists of amazing experts from across the globe, and they are going to be sharing with us the impacts of pollution on the deep sea, the processes which gets those pollutants into the deep sea, and some of the policy frameworks that can be used and have been used to address these issues. So if you've got any questions for our amazing speakers, then please use the Q&A function at the bottom of your screen. If you'd like to talk amongst yourselves, then please use the chat. And it's really important that everybody knows that this session today is being recorded. If you do not want to be recorded, then do not make any comments on this forum. At the end, we'll be asking you to do a survey, and that will be about your takeaway your takeaway message. That'll be after the Q&A. So we're through with the introductions, and I have the immense privilege and pleasure to introduce all of our speakers today. Our first speaker is going to be talking to us about pollution in the deep sea. His name is Ivan Hernandez, and he's a recent graduate from the University of Gibraltar, where he's undertaken his MSc in Marine Science and Climate Change. And he's going to continue his work on marine litter, its accumulation and distribution during his PhD in Barcelona. So Ivan, over to you.
Hello, everyone, and thank you very much for the introduction. I will now share my screen. Okay, so I'm going to be talking about an introduction or an overview on pollution in the deep sea. So as Lucy Woodall said, there are many different types of pollution, which are not limited to just plastics. There's sewage, raw sewage is still pumped into the sea in many places. That happens in Gibraltar, where I'm from. There's litter, which I will be talking about in a little bit. Oil seepage and spillage, which we've all seen the devastating consequences that can have. Fertilizers and pesticides, which can lead to, fertilizers especially can lead to eutrophication, which can cause algal blooms, and then oxygen depletion zones, which end up in huge die-offs. Noise affects cetaceans, and that is an active area of research, I believe. And then industrial waste and toxic chemicals. To focus a little bit on litter, the definition of litter by the United Nations Environmental Programme is any persistent, manufactured or processed solid material which can be found in the coastal environment. So things like vegetation that can sometimes be seen after severe weather events, that wouldn't be considered litter as such because it decomposes. So litter can be classified by composition. This can be plastics, fishing material, metals, construction material, even paper and cardboard has been found in coastal environments. However, these are not standardized. So even though there are protocols, these are rarely followed. And many studies just follow their own guidelines on on litter composition. So if we focus a little bit from litter onto plastics, we've seen that the production has increased from about 5 million tons in the 1950s to over 350 million tons as of 2015. I think that the current figure stands close to 400 million tons as of now. And the amount of plastic in the ocean has grown, or is estimated to grow 300%, up to 150 million tons by 2025. And 90% of this is believed to come from just 10 rivers located in Asia and Africa. However, without wanting to point any fingers, this has got to do with improper waste management, and it's not a problem just of developing nations. Developed nations also export a lot of their litter to these other countries. So we all have a responsibility to limit the amount of litter that gets into the marine environment. And there's believed to be about 5.25 trillion plastic particles floating on the sea surface alone. These are particles greater than 5 millimeters, corresponding to about 240,000 tons. The thing about plastic is that they're not all the same, as we all know, and the buoyancy is highly variable. It depends on the density of the and the composition of the plastic item. So about 50% is believed to, that it will sink quickly after it enters the marine environment. 35% is believed that it will float for potentially great distances before sinking. And the thing is, most of the time, we don't know where an item of litter or plastic has entered the marine environment, and we don't know how far it has traveled. So we don't really know what the sinking rates are either. And about 15% will stay floating or in the water column, so that is between the surface and the seabed. And the thing is, density of the plastics also changes with time. And this can happen because of biofouling, which means that bacteria and algae will grow on the plastic item, and it will change density and can start to sink. Also, there'll be leaching of additives. I mean, plastics are added these chemicals in order to make them even more durable. But in seawater conditions, these can leach out and the plastics can become waterlogged, and then they can start to sink, or they can amass epifauna, so it can have organisms growing on them. Again, it will change density and it can start to sink down. Plastics or litter in general, can be classified also in terms of size. So macroplastic or macro litter is particles or items over 200 millimeters, and all the way down to nanoplastics, which are smaller than 1 micrometer, which is 1,000th of a millimeter. Modeling shows that 1 200 millimeter plastic particle can degrade the thing is, plastic does not decompose, it degrades into smaller particles. So one 200 millimeter plastic particle can degrade into 62,500 particles of less than a millimeter in diameter. In real life conditions, it doesn't happen uniformly. So you could get, orders of magnitude greater amount of smaller, even smaller particles being released. And the degradation of plastics on the surface occurs via photooxidation, which means that it requires ultraviolet light and is temperature dependent. So in warm climates, tropical beaches, this will be much greater than in polar climates, for example. And also biodegradation, which is done by bacteria. However, this is much slower than photooxidation, even in optimal conditions. However, this is just on the surface. On the surface, longevity estimates can be anything between 400 and 1,000 years. However, in the deep, this is, again, orders of magnitude higher because there is no light. Temperatures are very close to 0 degrees, and there is little oxygen that can be used for biodegradation anyway. So even though forever is not a very scientific term, it is estimated that items of plastic that enter the deep sea will stay there for hundreds of thousands, if not millions of years. So we also have to look at the properties of plastics. So they're considered to be biologically inert. They don't interact with organisms, which could be considered a good thing. They are hydrophobic, water repelling, which is why they are so useful to us, but they're also lipophilic, which means that they attract organic, especially oily organic compounds. Now, some of these oily organic compounds are known as persistent organic pollutants. Which are toxic chemicals which can accumulate inside organisms. And these can become, what happens is that these toxic chemicals, they absorb or they adhere onto the surface of the plastic item, and it can become up to 1 million times more concentrated on the particle than in the surrounding seawater. So the smaller the particle of plastic, the higher the surface area to volume ratio. I'm sure many of us have heard of microplastics. And the reason is that if you have, in the example I mentioned earlier, 62,500 items of microplastics, they will have, again, orders of magnitude greater concentration of these persistent organic pollutants and other toxic chemicals than one single large item of plastic. And microplastics have been considered to be a likely vector for the transport of pollutants into the deep sea. Nanoplastics can actually, they're smaller than microplastics, and they have an active area of research now because it has been observed that they can cross cell membranes and lead to cumulative toxicity inside cells. So plastics are everywhere, and there is a very high variability as to the accumulation of microplastics in the deep sea. Seafloor features such as submarine canyons are well-known things for litter, trenches as well. And there are various different impacts. Macroplastics, for example, will impact sessile organisms, so organisms that don't necessarily move around, such as sponges and corals, ingestion, suffocation, physical or mechanical damage, ghost fishing, which I believe will be talked about later on, even necrosis and death. But deep sea sediments have been shown to have over 4 orders of magnitude a greater concentration of microplastic particles than contaminated seawater. And it is these microplastic particles that can lead to ingestion and accumulation of toxic chemicals, which have been observed in various phyla in the deep sea. So what I've tried, even though this webinar is about beyond plastics, I believe that plastics are inextricably linked to other types of pollution. That is why I've tried to link the two. And I believe that this is a good place to leave this introduction, because I think other speakers are going to touch upon some of the points that I have made. So thank you very much for your time.
Wonderful, thank you so much, Ivan. You gave us a great summary and introduction to our wonderful speakers that come after you. So next, it is my pleasure to introduce to you Professor Charitha Pattaratchi. He's a professor at coastal oceanography at the Ocean Graduate School and Oceans Institute at the University of Western Australia. His research encompasses coastal ocean physical processes and their influence on the climate, biological and geological processes in estuaries, the nearshore and the continental shelf. And we are going to talk, he's going to talk to us today on the pathways for pollution and how they enter the deep sea. So Chari, over to you. Chari, please put on your video and unmute.
I hope you can hear me and see my screen.
Yes.
Okay, thank you. And thank you for the invitation to present. So I'm going to really, in 10 minutes, it's very hard to cover pathways to pollution. So I'm basically going to talk about microplastics. And I would like to acknowledge some of the work that was contributed to this talk by Dr. Vijayaratne. So on the left, you actually see a glider, and that's 1,000 meters. And unfortunately, that's one of the ones that I have lost. We found it, but we couldn't recover. So there is a, how do you say, human pollution too, and it's not plastic. So when we look at the deep sea, we think about pictures like this. This is I took last year in about 4,000 meters of water in the Bremer Canyon along the south coast of Australia. And in all my explorations, which is using the Schmidt Institute of Oceanography research vessels, we have hardly seen any plastics or litter around the canyons on the west and the south coast of Australia, which is really good. And that's what we really want to keep it to. So, as Ivan mentioned, plastics come from land, from human use, and it enters the ocean to rivers. I don't know how many of you actually know how the plastics actually started. It started in the mid-18th century when the game of billiards was very popular. But what was happening is that the billiard balls were made by ivory. Means that they had to kill a lot of elephants to make billiard balls. So a billiard manufacturer in New York came up with a competition to make a substance, an artificial billiard balls to conserve the elephants that they could make the billiard balls with. And the first substance that somebody came around was called cellulite. And unfortunately, it was very explosive. So, you know, billiard balls hit each other and they started exploding. So that actually stopped the making of the initial foray to making plastics. But come the 19th century, the 20th century, during the World Wars, the development of plastics came quite a lot. And most of the plastics is actually made from hydrocarbons. So what I'm going to talk about really is about how if this is just a schematic, that we have input from a river. And remember that microplastics that we're talking about is mainly floating. So about 35% of all plastics made globally have densities which are higher than seawater. And if they get to the ocean, they will sink. But 65% is actually buoyant material. And the only way microplastics can get to the deep ocean is that if their density changes through, let's say, biofouling or something like that, they can sink. Otherwise, they will basically float along the sea surface until they probably somewhere make landfall. But we know, we all heard about garbage patches, which is where all of the plastics will ultimately end up in the ocean unless they end up on beaches. But there is a whole lot of hurdles for that plastics to actually come across. So if you come from a river, you will first come encounter a plume. And all of these areas are what we call convergence areas, where the water comes together and sinks, and everything actually floats at the surface. So if you go on an estuary or a river plume, you normally see some foam or twigs and pieces of leaves, which are along the line, or there's lots of plastics there too. But the only thing is that those areas are also highly productive. So when you have organisms which are feeding on those frontal systems, you would have a lot of plastics ingested. So when you come out of the plume, you will come to ocean and oceanic, coastal and oceanic fronts. And then you actually come into the wide open ocean where the winds apply, the waves are active, and then the ocean currents, and ultimately, maybe many decades later, they will end up on garbage patches, or they will end up on a beach somewhere. So here you see a cross-section of a front. So you actually have the convergence of water at these fronts. The water sinks, but anything which is floating will remain at the surface. And that's really what we call the high system. So they can be destroyed by wind and storm systems, but often they remain in place for days to weeks. So here I'm now again showing you, forget about all of the observation ones. So if you've got a river coming in, this is full of plastics, you will have a river plume. You have wind stress, which is breaking on it. You may have the Stokes drifts. There is the surface Ekman spiral, and then you have Langmuir circulations, and all of those will basically end up of the water going from the river to the shelf region to the offshore. So here is a simulation of plastics, which will actually end up on the ocean. On the left is what, if you start off with no plastics on the ocean at all, after 1 year, this is what you will see from those global riverine inputs. And then after 10 years, you'll see that they're all ended up in the middle of the oceans through the different garbage patches. So in all of these, main thing with the plastics is they would actually remain on the surface and transporting with the waves and the currents, and they will accumulate in the center of the oceans or ocean basins, or they will end up on a beach. But I'm going to show you an accident, which is the Express Pearl, which happened off Sri Lanka. Around here, it originated from Qatar, sorry, in Dubai, went to Qatar, went to India, and came. And basically, in early June, actually had explosion, and that was the actual ship, and it sunk off Sri Lanka when it caught fire on the 2nd of June. So at this time, we basically have the currents as such that we basically have currents going around Sri Lanka. We have the Sri Lankan dome, we have onshore winds, and the waves from the southwest. And then, and one of the things that he actually had, you will not see this all that well, but I wanted to just hold on, it'll come better. And this is what happened on the beaches. These are microplastics, which we call nurdles, and they are the raw material that makes up most of the plastic. So your toothbrushes, your plastic bags, and all of them basically is made up of these nurdles. And all of this basically ended up on the beaches. You can see the ship on fire in the background, and now you actually have. So here, there was 78,000 tons, I'm going to come back, 78,000 tons of nurdles. A recent estimate says that there's 6,000 tons per year comes from all of the global rivers of the microplastics. So 6,000 tons is combined for all of the global rivers, and this one event released 78,000 tons of nurdles. So there's a cleanup going on, and then you basically had some of the fish which was ingesting. You can see the nurdles inside these systems. So here you actually see a nurdle wave on the beach. So now I want to try to show you how these nurdles then get transferred. So this is actually comes from the coast. The red and the white dots are basically we're tracking those nurdles through the water to the surface water, and with the system that we have to predict. So, and then I'm going to show you this, but then after a few days, you actually see the back, you see the arrows, so the currents, and the color, the background color is the temperature. So around here we have upwelling, and you can see that this water remained, and the plastics basically remain in this system. So you can see this front here, between the warmer and the cold water, and you actually see this is holding it up. And then what happened was that this is now, I'm going to show you a longer simulation. So you actually see the plastics coming here. Now there's internal waves which get generated through the Andaman Sea. You can see these stripes in the vertical, and as this plastic comes around, so now we're talking, look at on the top, this is after about 3 weeks after the spill, and then you actually end up in about a month now, coming on to the 25th, and you see on the other side, onto the west, is going back. Now this is the southwest monsoon, and at the moment we'll be coming to the end. So all of these currents will reverse, and all of this material here will come past the Maldives and then go towards Somalia. Some of this will end up in Indonesia as well. So this one event is going to cover the whole of the northern Indian Ocean, and these plastics are going to be on the surface for many years to come. And part of this also, we didn't expect, you can see here, the plastics went to the Gulf of Mannar here to the north, and that's because of a cyclone which came through here, and that created a wave which went all the way across Sri Lanka and pushed all of the plastics back across to India. So we can follow this. We had a citizen science which was run by Asha DeVos, where people went and took photographs of the nurdles, and we could actually cover by observations along the beach with the nurdles and validate our models. So in the summarize, there are many pathways for buoyant material to enter the deep ocean. The ocean dynamics are really, really important in controlling all of these systems. Thank you.
Charitha, thank you so much. I really love the way that you told us about ocean processes, but gave us that really unique example coupled with the citizen science. That's a really powerful case study to share with us. So thank you for your time today. Our next speaker is going to talk to us about the impact of ghost fishing gear on marine life. Her name is Elizabeth Hogan, and she is a senior program manager for the National Geographic Society. And she's worked on ocean plastic pollution with NGOs from across the globe for over 10 years. She's a specialist in marine wildlife, entanglement and sustainable fisheries. So Elizabeth, over to you.
Hello, everyone, and thank you so much for the introduction. Lucy, can you all see my screen?
Yes, we can. Excellent,
thank you. Yeah, as Lucy mentioned, I am here to discuss the impact of fishing gear on the deep sea. The lost and abandoned fishing gear is most frequently referred to as ghost gear, and that's a term that you'll hear me use throughout the presentation. Its other name is abandoned, lost, and discarded fishing gear, which is the horrible acronym ALDFG, although that is how it is frequently referred to in policy documents. Ghost gear has only recently, and by recently I mean within the last 6 years, become an item of ocean pollution that has been discussed or even acknowledged. And as such, its effect on the deep ocean is something that we are only just beginning to understand. Most of the work done on lost and abandoned fishing gear has been done in shallower and coastal waters. And so we are now in the position of extrapolating that data to learn what we can about its effects on the deep ocean. The estimate is that approximately 800,000 tons of gear is lost in the oceans every year. That number is based on the 2015 JAMBEC study, and combined with a statistic on gear loss from the FAO, the number is likely very low. And as other speakers have referenced, once gear is lost in the marine environment, it can move around pretty much wherever it likes, and it will last for hundreds and hundreds of years, continuing to cause damage. It's important to remember that fishing gear is not like other plastic or metal lost in the ocean. It was literally designed to trap and kill wildlife, and once lost, that is exactly what it continues to do. When you think that only 27% of ship waste is properly disposed of, it does make you wonder, where does the rest of it go? It has to go somewhere. And to picture that, you need to think in terms of the finite capacity of a ship. While intentional discard is often affiliated with illegal fishing, and it is more likely to come from illegal fishing, you also have to consider the fact that if a fishing boat has room on board for extra catch or room on board for gear that no longer works properly, there is some incentive to dispose of gear at sea, especially on the open ocean where there is no enforceable regulation. The estimate is that approximately 136,000 marine mammals are injured and killed from the gear each year. Mammals really here only referring to cetaceans and pinnipeds, so whale and dolphin and seal and sea lion species. And millions of sea turtles and fish are also lost to ghost fishing each year. And when you think of nets and how they function, and how fish are just as likely to swim into a ghost net as to one that is actively fishing, consider the effect that that's likely to have. A fish is dead, a predator smells it, they approach the net, and then that animal is likewise trapped. And so the nets present this ongoing pattern of trapping and killing wildlife. Here are some stats that I am sharing with you on ghost fishing, not to inform you on the statistics, but more to emphasize the difference across species and geography. Gear moves around all over the world, as I mentioned, based primarily on ocean currents, tidal movement, and turbidity, and it creates hotspots. So some stocks and some ecosystems are impacted more than others. If you look at the very bottom photo here, it represents the interesting case of the hotspot of Hawaii. There are no net fisheries in Hawaii, only longline fisheries, and yet tons and tons of nets wash up on those shores every single year. And the Hawaii program for deep sea net removal is the most advanced in the world. And every year they pull between 50 and 60 tons out of the ocean. That's just one single snapshot of some of the nets that they remove on an annual basis, which means that the same amount of nets keep returning to the same spot. If you look at the top image, you will see a photo taken from the Marianas Trench, where plastic bags and fishing gear were found on the most recent expedition. So we do know that fishing gear makes it to the deepest point of the ocean. And if you look at the middle photograph, those are identification tags from lobster traps from Canada and the United States, from the northeastern coast, Newfoundland, Maine, and New Hampshire of Canada and the United States. Those tags and the traps that they were around were found on the east coast of the United Kingdom, meaning that the traps, which are metal coated in plastic and designed to sit on the seafloor, made it all the way across the Atlantic Ocean. So we know that the seafloor is definitely affected and that this gear can travel pretty much wherever it wants to go. Ghost gear is not just impacting large animals, as I mentioned, it's also impacting entire marine ecosystems, and in particular coral reefs, which are affected by active fishing, yes, particularly bottom trawling, which, as we all know, has the capacity to crush and remove valuable coral from the seafloor. But the lost gear also impacts these coral ecosystems, both in shallower tropical settings as well as deeper cold water coral ecosystems, both of which are pictured here. And as you can see, there was a wonderful study done in the Sea of Oman, which assessed the damage caused to coral reefs, of half of them harmed by ghost nets, and ghost fishing making up the overwhelming majority of the anthropogenic impacts on those coral populations. And the most prominent effects of the plastic litter as well and ghost nets on the seafloor in that arena tend to cause the smothering and create the anoxic conditions that will reduce production rates, respiration rates, and filtration rates of the sea life and ecosystems. This is what's called a fish aggregation device. It's known as a FAD, and they are most commonly used to catch tuna. As you can see, the structure of the gear is a raft or floating platform, which hangs nets through the water column to very deep levels. This one obviously fits on camera. One of the challenges with depicting the impacts of ghost gear is being able to capture the image of how large a net can be. Many FADs will be up to a mile in length, and they can extend vertically for hundreds of meters down into the water column. In order to do that, they have to be weighted. So when they become detached from the rafting platform, where do you think they go? They will immediately be dropped down onto the sea floor. And not only are these used to catch millions and millions of tons of tuna per year, but they also catch over 100,000 tons of bycatch. So a great deal of marine life impacted by FAD fishing. In the next slide, you'll get a bit of an image of how much of the ocean they can cover. But when the ocean cleanup did their assessment of the North Pacific garbage patch, they discovered that 46% of the debris in the North Pacific is actually fishing gear. And as I mentioned, because they already hang so deeply into the water column and they are weighted, their capacity to sink and impact deep ocean ecosystems is tremendous. So this is a satellite image, just one singular view of the FADs, just FADs. There's other fishing gear there as well, of the Pacific, particularly the Coral Triangle, where tuna is most commonly caught. So imagine what it would be like for a whale to be swimming through this, trying to avoid this extent of nets. Each green dot that you see represents a single FAD. Those FADs do have transponders attached to them so that the seafood company which owns the gear is able to collect it when the weight suggests that there's enough fish to collect. However, within the seafood industry, it's not uncommon for a free-floating FAD to go so far out of range that it isn't worth the fuel cost to go and collect it, and therefore the transponder is simply switched off, and that raft and those sets of nets are left drifting in the ocean. So it creates the capacity for a significant amount of ghost gear to float throughout. This is an image of a sperm whale, mother and calf, unfortunately caught in the exact same net. Sperm whales dive fairly deeply to about 2,000 meters, so their capacity to drag nets with them and transport the nets is not to be underestimated. This was a particularly sad case where a rescue team was alerted about the condition of the animals, which are mammals and therefore have to surface to breathe air. Unfortunately, the calf died, although the rescue team was able to save the mother. This is just a very quick clip that I'm going to show you that gives you an idea of what the rescuing of, or disentanglement rather, of a whale in the open ocean is like. It's only 9 seconds, but this is the work that has to be done to try to remove lines and nets from these animals. Not an easy thing to do at all, but again, at their size, their capacity to become entangled and then move nets, and of course any animal fortunate enough to be disentangled, the gear then it's recovered if the team is able to recover it, but sometimes that's not the case, and the gear can then also be dragged further by the whale or simply dropped to the sea floor. There we go. So these are the removal options that currently exist. However, they all obviously function in much shallower waters. In the upper right, you'll see a barge and winch methodology that's used in the Gulf of Maine. Those are some wonderful fishermen who also happen to be divers that were willing to recover some of this gear. And you'll see a great deal of metal mixed in. That was simply one batch of gear that was pulled up that day off of those fishing grounds. But obviously you can only remove it for as far as you're able to hook and haul the gear up onto a barge platform. In the lower left, you have divers in the Caribbean. A wonderful group from various Caribbean countries came together to form a network to collaborate on ghost gear removal. Divers with pulley systems were able to remove a great deal of gear on multiple occasions working together. But again, that can only function as far as divers are able to go. The middle photo on the bottom represents one of the systems worked on by the divers in Hawaii, who again have one of the best removal systems out there. And you'll see they and the photo on the right, the Ghost Fishing organization here operating off the coast of Scotland, use float bags to dive and attach to the gear and bring it to the surface that way. But all of these methods are again designed for shallower waters. And since the ghost fishing problem has only been known to us for a few years, and we've worked out solutions for shallower and coastal waters, the next challenge that lies ahead of us is to establish removal methods that will work in deeper waters where it's much harder to get visibility and ascertain where the gear is located. High kilohertz sonar scanning can be helpful, but once there's no light, that too is a very difficult proposition. And many removal methods right now are quite expensive, considering the technology required and specialization of those who can conduct such removals. So that is without a doubt the next step in the journey for removing this material from the deep ocean. And thank you all very much for your time.
Elizabeth, thank you so much. I think your stats and your images are incredibly powerful to show us where the challenges are and the challenges that we know about. And I think your note there on the end about what we still don't know and what we're only just discovering is a really powerful aspect to leave us on. So thank you so much for your time today.
Thank you.
I now have the pleasure to introduce to you a researcher from the University from Chile. This is Patricia Zarache, and she's an expert on sea turtles and large pelagic animals. Her field of research is involved looking at the population dynamics and applied ecology for marine systems conservation and fisheries management. She's going to be talking to us today about the highly migratory species, fisheries, and the ingestion of plastics and heavy metal and biomagnification. So Patricia, over to you.
Hi. Hello, can you hear me well?
We can hear you, thank you, and we can see your slides.
Okay, great. So, well, good morning, everyone. Thanks for the invitation to participate in this interesting webinar. Today, I'm going to talk about the research we carry out in the Southeast Pacific Ocean, particularly in Chile. We work at the Instituto de Fomento Pesquero. From now on, I will refer to it as IFOP for its initials in Spanish, it's easier. IFOP is the official advisor of the Chilean government in marine science and fisheries. And I will also be highlighting the work from other researchers from Chile along the talk. So let me give you a brief overview of the fisheries we study. The dots in the map represent fishing sets. That is the event when the fishing gear is placed in the water to capture fishes. And the colors represent different fisheries. So we hear three different types of fisheries. Two using long lines of different dimensions, and the other one using nets that are called gillnets. So we can see that these three fisheries uses a large geographic area coming from 18 degrees south to 40 degrees south, which is a very, very long area in Chile. And these fisheries targeting swordfish, which is called albacora locally, and also targets sharks and mahi-mahi or dolphin fish. But we also study the ecology and the biology of bycatch organisms, those who get caught incidentally in the fishing gear, such as sea turtles, marine birds, marine mammals. And they are considered as protected species and also as threatened species. So they are facing an extinction due to human activities. The species we study are organisms known as highly migratory because they travel thousands of kilometers between their foraging and reproductive grounds. In the figure at your left, we see the movement of fishes, sea lion, albatross, sea turtles, and sharks. So they travel across the ocean to reach these critical habitats. But they also undertake long and deep immersions during the day, searching for food. So they inhabit the pelagic ecosystem. They live in the first 100 meters of the water column, but others make deeper dives through the mesopelagic zone below 100 meters and even go farther down below 1,000 meters, such as the whales and sperm whales. So during this movement, when they travel across the ocean or take this immersion after they prey, they face several threats that affect their survival. Plastic debris is one of the most important threats affecting marine mammals. Here we see at your left a map based on 2 research cruises. These cruises were carried out during 2015 and 2016, and they register the amount and location of the floating plastic along the route of the oceanic islands of Chile, including the Juan Hernandez archipelago in the bottom, and the Desventuradas Island on the top, and then to your left, through Salas y Gomez Island and Easter Island, called also as Rapa Nui. This study concluded that the highest abundant and high density of litter, plastic litter, was located near Easter Island and Salas y Gomez, because here there is this subgyre in the middle of the Pacific. So the litter is aggregated in the near shore of different countries, and then is pushed through currents or by the currents to oceanic waters. So at your right, we can see another study where they concluded that the ingestion of plastic by marine fishes also is aggregated in higher proportions in the same area. So these 2 studies concluded that we could be seeing more plastic ingestion in species that are in the oceanic water compared to the one in the coastal waters. So here we have all this plastic around this area, but it's the same area where fishes, we can say sharks, we can say pelagic fishes, and also turtles move around. So here we can see the connectivity within the Pacific Ocean of different turtle species. They move across the ocean from north to south, from east to west, and the other way around. And we also see sharks moving in different areas of the same ocean. So the fisheries observers from IFOP regularly collect biological data and samples from these pelagic species. They get on board the longline and gillnet fleets and collect different type of information. Since the 2005, we have been studying the diet of the pelagic ecosystem, but just recently we started focusing in plastic litter inside the stomach of these animals. So during 2019, we observed mesoplastic in the stomach of one specimen of mahi-mahi, or dolphin fish, in a coastal area. The plastic that we found was associated to fishing practices. It was a tide cable normally used by fishermen. And the ingestion of plastic debris by mahi-mahi has been recorded across the ocean of the world, not only in this area. suggesting that there is a non-selective and voracious feeding behavior of this fish. So this is what drives the plastic intake for the specimen. So this species is an opportunistic predator. It could eat what comes in front of it. So this increased the risk of plastic ingestion. Plastic items are probably confused with prey, or they ate a prey that has the plastic inside its body. It could be either both. However, further studies are needed to confirm this. Other records of plastic intake comes from the blue shark, one of the most abundant species in Chilean waters, but the proportion of stomach with plastic was very low. It was only 1.4% of the stomach with a plastic content. Other records describe the presence of bottle and pen caps in other areas of the Pacific. And this low presence of plastic was also observed in Peru and some other areas in Canada, in the South Atlantic, but researchers in the Mediterranean found more stomach with plastics, but this could be reflecting the amount of litter in the specific areas. So blue sharks ate or eat in the upper part of the water, more near the sea surface. So it is not surprising they could eat floating plastic bags. Higher records of plastic were found in other species. This is the opa, or called sunfish. And we found plastic intake in several years, and the size of their fragments found was larger compared to the ones that we found on the blue sharks and in the mahi-mahi. And part of the plastic was identified as fishing gear, and also there were lots of plastic bags, and the fishes were located near Oceanic Island. This species has shown a high incidence of plastic ingestion in other places where more than 40% of the stomach has plastic. What is intriguing is that the opa is a mesopelagic fish living in waters below the 100 meters. So why a mesopelagic predatory fish are feeding on plastic debris of a presumably surface ocean origin? So a few theories remains in place. One is that the fish could move up to the surface and feed, but if that is the case, there will be more records, or the litter can also be found in deeper waters, and this seems to be the case. If we look at another species, if we look at sea turtles from the Southeast Pacific Oceans, we can see that litter ingestion and entanglement in plastic debris have been recognized as a serious threat to this species worldwide, because they tend to spend longer time at the surface, where they encounter drifting fishing debris and litter. Stranded turtles that we found in different locations along the north and central Chile, and also in Rapa Nui, were transported to facilities. They were treated. Some of them spent months in this facility, but most of them did not make it, did not survive. The microplastic found in their stomach came from different sources. Some of them were plastic bags, monofilament, nylon, and rope, and fishing nets, all from fishing practices. And the plastic injection causes blockages in their guts, or even perforation, and the eventual death of this animal. So sea turtles are among the first data recorded to ingest plastic debris. So all these biological samples that have been collected over a period of 16 years by our fisheries observers have helped us to build this pelagic ecosystem food web. So the first years we studied only stomach contents of top predators, and eventually we start adding in the prey items and also stable isotopes and fatty acid analysis, so we came up with this very simplified pelagic ecosystem food web that is based on those analyses. And in here we can see some of the predator-prey interactions. So here is who is who. For instance, we can see that some sharks eat dolphins. So here we have dolphins are eaten by some of the sharks. Some sharks also eat sea turtles, and some sharks can eat cephalopods or can eat different types of fishes. So what happens if one of the small fishes here in the bottom absorbs a toxic element from the environment. Due to the interaction of this food web, that toxic element will reach up to the top predator of the ecosystem. And that is what is called biomagnification. So when a non-degradable substances increase in the ecosystems, and they increase the concentration of the toxic substances among the organisms present in the food chain and food web. So the poisonous level gradually increase with the increase of the trophic levels of the food chains. This means that the top predators of the ecosystem, whether they are marine mammals, fishes, or sharks, they will have a higher concentration of the toxic element in their body. That can lead to a human diseases because humans eat the fish, and in certain countries, they sadly still eat cetaceans. So we have some preliminary data regarding
Patricia, can you just give us one more minute, please, because we've got to move on to our next speaker. Thank you.
So I'm going to show you here what we found. So we have cadmium, mercury, and lead. And we see that for cadmium and lead, we have higher levels compared to what is safety for human health. And when we see sharks, we also find that lead is also higher in the meat compared to the safety levels. And when we look at heavy metal concentration in sea turtles, we also have found copper and lead, and all values way above safe levels for human health. And the other find that we have is the first record of fibropapillomatosis in a sea turtle, which is very related to environmental health and pollution in different type of waters. So this animal died after 19 months period of rehabilitation. So this is still a work in progress, and we definitely need to improve the sample size, but some general conclusion can be drawn. So we can see that there is plastic injection in fishes that we commonly use as source of protein, and also in threatened species. This plastic pollution continues to extend into the different zones in the ocean, into the deep ocean. And the concentration of heavy metals is also in sharks and swordfish, and is a hazard for human health. And the determination of these current heavy metals in this species needs to be updated because they are from the 2011. So we need to know what the levels are at the moment. And the presence of fibropapillomatosis recalls for a more detailed examination of the individuals, because this disease can potentially spread to other sea turtles. So thank you very much. And I would like to just acknowledge to all observers on the field and to the permits given by the authorities. Thank you so much for your attention.
Patricia, thank you so much. You had some really interesting insights from a really unique part of the world, and being able to share with us actually the consequences of some of these pollutants on human health is incredibly powerful. Our next speaker today is Shamik Gupta. I've already introduced him once, as he's a co-lead of our working group today. He's an associate professor at the Institute of Deep Sea Science and Engineering at the Chinese Academy of Sciences. He's involved in the exploration of deep sea pollutants, and he's going to be talking to us today about the pollutants that were found in the Marianas Trench. Shamik, over to you. Thank you.
Thank you, Lucy, for the introduction. Can you see my slides and hear me?
We can see and hear you, thank you.
Okay, thank you. Since we are running out of time, I'll try to keep it short and to the point. So today I'm going to talk about the anthropogenic organic pollutants, rather the persistent organic pollutants in the Mariana Trench sediments. Before we begin the main presentation, just a few interesting and some interesting points about our ocean and the deep ocean. Some things that many of you might not know, that the Pacific Ocean is actually wider than the moon. For example, from in its widest part from Indonesia to Colombia, actually it's like 5 times the diameter is more than that of the moon. And the ocean is home to nearly 95% of all life. So it consists of a major portion of life that we have in the earth. And the pressure, as you know, is huge in the bottom of the ocean. And more people have been to the moon than to the Mariana Trench. There is something interesting. And oxygen, no, atmosphere is not the greatest source, but our ocean is the greatest source of oxygen. And we have better maps of Mars than we have of the deep ocean. So today I'm going to talk about the anthropogenic pollutants in the Mariana Trench. Just to give you an idea about what is the Mariana Trench, how deep it is. So as you can see from the picture in the left, it is more than 11,000 meters deep. And if you consider the highest mountain range, Mount Everest, you can fit a whole Mount Everest inside the Mariana Trench. It's in the Western Pacific Ocean. And the first people who went down to the Mariana Trench in a manned submersible were Don Walsh and Jack Pickard many years back in the 1960s. So for us, we collected sediment samples from this place highlighted in the southern part of the Mariana Arc, as you can see. We collected a number of sediment samples and did the analysis on 5 of those samples from the bottom of the Mariana Trench, ranging from 7,000 meters to more than 10,000 meters. And we tried to extract the POPs, like the persistent organic pollutants, and see their concentration and how they vary with the depth and with other parameters. So before I give you the results of what we find, just a brief introduction about what persistent organic pollutants are, also called POPs. So basically, these are organic chemicals, synthetic organic chemicals, as you all already might know, and they are persistent from the names. They stick into the environment for a really, really long time, and it's very difficult to degrade. And they can travel really long path in the ocean, and that causes a global pollution. They are lipophilic. They love fat, so they can get attached to fat very fast and very easily and stay there for a long time. And they bioaccumulate in the food chain, as in the last presentation, Patricia said that she talked about biomagnification. So this is one of the things that biomagnifies in the food chain. As we go up in the trophic level, the concentration increases. So we already found very high levels in the fishes and marine mammals, and it's toxic. It's very much toxic and can cause serious problems in our systems. It includes such chemicals like PCBs, like polychlorinated biphenyls or PBDs, or for example, pesticides, DDTs, and all those chemicals, very harmful. So POPs in the Mariana Trench, we found high concentration of POPs in the sediments. And also before us, there was one study by Jamison, and they also found very high concentration of POPs in the Mariana, in the amphipods, in the endemic amphipods. These kind of shrimp-like creatures in the trench from 7,000 to 10,000 meter, they found high concentration of POPs in such a great depth. And the concentrations were greater than the baseline levels and almost 50 times greater than in crabs from a very highly polluted river in China. So that is quite amazing, because when we go to such great depth, like 10,000 meter, we expect to see a clean environment. We expect to see a very neat pollution-free environment, but it is not so. It is not what we thought. And when we measured the POPs concentration, the PCB concentration specifically in the Mariana sediments, we found that the concentration is very high. So left over here, it's the picogram per gram. We can see that in all these 5 samples, the concentration ranges from about 930 to more than 4,000 picogram per gram. So what does it mean in terms of in comparing to other areas? So if we compare the concentration of the POPs in the deepest Mariana Trench sediments to some other sediments across the globe, so we find that our data lies in the high area, around 4,000 to 5,000 picogram per gram, whereas these samples, which were collected from surface sediments from these places like Gulf of Alaska, Lyon in the Mediterranean region, in the Bering Sea, in the Icelandic station, all of them, the depth of water is much less than the Mariana Trench, but their concentration of POPs is also much less. So it's amazing that how much polluted the deepest trenches are right now. So how toxic is it in terms of causing harm to the heddle population? So we try to find the toxic equivalence of these PCBs. There is a process to do that, to multiply 12 dioxin-like PCBs, which are the most toxic, with some factors called the HU-TEF, toxic equivalent factors, and we get some numbers. And then these numbers indicate how toxic these kind of samples, the sediments are, when we consider the concentration of the PCBs. So we find a number over here, which is 0.65 to 14.87 picogram per gram. And then again, when we compare this number to some areas which are industrial areas or semi-industrial areas, or even highly polluted river, like in Han River of Korea, or in the Central African, South Africa, we find that the Mariana Trench, the sediment toxicity is actually quite high compared to most of the polluted industrial or semi-industrial areas. So this is something, again, we need to worry. So just the take-home points, a few important points, that the heddle trenches that we thought are clean are no longer clean, and man-made pollutants have actually reached the deepest point on the earth. So how are these pollutants accumulating in these trenches, in these deep trenches? So there are different assumptions and hypotheses we think that this could happen. One of them is the post-mortal discharge, when the heddle amphipods, they died, then the pollutants they release from after their death, it accumulates into the sediments from the wax esters, or also they can release from the ingestion and fecal release of these kind of amphipods. And also it depends on the morphology of the trench, on the geotectonic activities inside the trench, the V-shaped funnel-like shape of the trenches, and the fluid dynamics, and the occasional earthquakes and landslides inside the trenches. They all aid in concentrating this kind of pollutants in the deepest places. Also, before, we always had the idea that organic matter is the main thing that actually holds up, actually absorbs this kind of pollutants. But here we find in the Mariana sediments from some XRD studies that we have a lot of clay minerals. So we assume that maybe the clay minerals play a major role in the absorption of this kind of contaminant particles in the Mariana Trench. So with that, I would like to end my talk, and let us understand and protect our oceans more, and hope you all are doing excellent. Thank you.
Shamik, thank you so much. Thank you for introducing us to the world of those deep sea trenches and those additional pollutants that we don't necessarily see in the photographs. We're now going to move into our final case study, and this is from Dr. Inês Martinez. She's a researcher at the University of Azores in Portugal. She's an interdisciplinary scientist with an interest in marine ecotoxicology and ecophysiology. Her main goal in her research is to identify the molecular mechanisms behind the control and detoxification of metal ions in the deep sea. And she is going to talk to us about these pollutants in her talk. Please go ahead, Inês, we can see your slides.
Hi, hello, good afternoon, everyone. Thank you for having me in this great webinar. Today, I will talk to you about the impacts of how copper can have on cold water corals under a predicted scenario of ocean acidification. So human activities have impacted the marine environment in many ways, like the burning fossil fuels, the overfishing, the plastic pollution, the seafloor exploitation. So these impacts have triggered climate change with huge consequence to the environment. So living in this changing planet, the deep sea organisms have been adapted their physiology to cope with such changes. So it is very important to evaluate and understand how resilient they can be under a scenario of cumulative effects of this climate change and the many human activities. So one of the human activities that have been impacted a lot, the deep sea organisms, are the deep sea mining on seafloor. So this exploitation of interesting polymetallic sulfide deposits that are very rich in base metals like iron, zinc, copper, gold, sulfides, and other elements are very important to the industry, and but they are trapped in that minerals and become available to the deep sea organisms when they are exploited at the deep sea. So not only these organisms have been impacted with these physical particles, but also from the dissolved metals that will be available in the water column. So in the Azores region, previous research has demonstrated the existence of a rich ecosystem of cold water corals in the vicinity of the industry exploitation areas. So we are very concerned of what the consequence of this predicted exploitation to these such as vulnerable ecosystems. So ocean acidification have been a problem in the last years because this amount of pollutants that releases the CO2 to the atmosphere the CO2 will be absorbed by the ocean, and this CO2 will change the chemistry of the seawater, became the seawater more acidic, and there is less available carbonate ions that are very much important to the good health of organisms that have shells and skeletons like cold water corals. In fact, ocean has become nearly 30% more acidic than it was at the beginning of industrial area. So it's a very huge problem. The Azores archipelago is located in the North Atlantic and is pretty much in the middle of these anthropogenic impacts. You have the North Atlantic gyre that is bringing a lot of plastic pollution to our shores, that this idea of the industry to explore the hydrothermal vents in the vicinity of the Mid-Atlantic ridge near the Azores archipelago. Also the ocean acidification that is predicted to occur also, of course, in the Atlantic Ocean. So why cold water corals are so important to study? So for the ones that don't know, cold water corals are the deep sea corals, the ones who live in the dark, in the cold waters, and they are pretty much ecosystem engineers because they harbor a lot of fishes and other invertebrates that depends on these ecosystems to protect themselves and also to find food. So they are particularly vulnerable to anthropogenic impacts and other environmental perturbation. So it's a perturbation not only to these specific ecosystems, but a bunch, all biota that depends on these specific ecosystems. In the Azores, these cold water corals are important components in our seamounts and our island slopes. So what we wanted to do, we already know that copper ions, like other metals, inflicts cell oxidative stress. They are very important ions to the function and homeostasis of cells, but when they are in excess, they bring a lot of oxidative stress to the cell. We also know that ocean acidification decreases the available carbonate ions for corals, like the cold water corals, that depends on these ions to form their skeletons. But what we don't know is how cold water corals respond to a predictive effect of copper exposure under an ocean acidification. So we perform an experiment, a land-based experiment, with two important components of the Azores cold water coral gardens. We use the seafan octocoral Dentomricea meteor and the whip-like octocoral, the Viminella flagellum. They occur pretty much at the same depth. So very shortly, we set up an experiment in 27 days, and we exposed the cold water corals to 4 different treatments. One of the treatments, we use the regular pH that we have at our days. Another treatment, we use that same pH, but with a concentration of copper known to be toxic for them, and also known to be one of the concentrations found in the release of mineral particles that we performed early on at our lab. And another one was an experiment when we use a low pH, predicted to be the pH of the seawater in 100 years. And the last one, we had a treatment with that low pH and with that acute concentration of copper. So we pretty much controlled the pH at the seawater by bubbling the seawater with either pure CO2 to decrease the pH or with CO2-free air to increase the pH. But I will not enter in details for now. So we analyzed a lot of parameters, very important to understand what happened to the cold water corals during the experiment, the dissolved oxygen. It is very important to understand how the dissolved oxygen decrease during the experiment to extrapolate the respiration rates of the corals. We also analyzed the copper in seawater and the tissues of corals to understand how they accumulate in their tissue and skeletons. The tissue loss was one of the parameters to understand how they die, what time they take to die during the experiment. And also the mucus is one of the excretion parameter when they are under stress. So very quickly, when we start to do the experiment in 27 days, we didn't expect it that started to die after 1 week in the copper treatments. So you can see in the pictures in the right place of the screen, they start to be necrosis here, very black, they lose tissue and also produce a lot of mucus. We use biomarkers to understand how they react to this experiment. We use metabolic detoxification of oxidative stress, damage, and response, and understood that each coral have a specific cellular response to cover this exposure to ocean, to low pH and copper exposure, but they are both very sensitive to such scenarios. Also, we wanted to analyze and identify the transcript biomarkers that were differently expressed between the exposed state and the healthy state. So to finish, turning up to the main question, what water coral responds to a predictive effect of copper exposure under ocean acidification. No, they have low respiration rates, the mortality in a short-term period, an increase of cell signaling for antioxidants defense, and the transcriptional change associated with copper exposure and ocean acidification. So although with physiological specificity, these both cold water corals are extremely sensitive to the cumulative impact of anthropogenic activities, and this much have to be included in environmental risk assessment when we are dealing with these anthropogenic effects. So thank you very much. I'll leave my email if you want to send me some emails to know more about this specific experiment or others that we have been doing with cold water corals. Thank you very much.
Yes, thank you so much for sharing your elegant experimental design for really exploring those multiple challenges that we have in the deep sea. Thank you so much. It's my pleasure now to introduce you to our final speaker today. She's going to talk to us about governance. Her name is Karen Robenheimer, and she's a lecturer at the University of Wollongong in Australia. Her research is on policies to prevent marine plastic pollution, has worked closely with the UN EP, Regional Seas Programme and governments across the globe on this issue of plastic pollution. She's going to talk to us today about the legal and policy frameworks to prevent plastic pollution in the deep sea. Karen, over to you.
Right, thank you, and hello to everyone. Thank you for the invitation to participate. I'll be providing a brief summary of the international and regional frameworks available to prevent plastic reaching the deep ocean. It's important to remember here that the implementation of these agreements takes place at the national level. So when countries sign up to these agreements, they commit to integrate the measures that are of that particular agreement into their own national legislation. So that's a key thing to remember as we go through the presentation. So just before we get to that, firstly, we've heard about some of the other types of pollution that can make their way to the deep oceans, and many of the pathways will apply to plastics as well. Some of these pathways are illustrated here, and this shows the complexities that policy must deal with, because each of these sources could have different drivers for leakage into the environment. So how is policy trying to deal with this complexity? Firstly, we currently distinguish between land-based sources of marine pollution, shown here in green, and sea-based sources, shown here in different shades of blue. Policy also distinguishes between areas within national jurisdiction and areas beyond national jurisdiction. This distinction is important in the level of control international and regional agreements can impose on countries. The Law of the Sea Convention is regarded as the overarching legally binding agreement that sets the foundations for the duty of countries to protect and conserve the oceans. It also defines jurisdictional zones, which are simplified in this diagram. When we talk about the deep oceans, they can, in theory, occur in any of these zones, and plastic waste can cross through any of these zones to reach the deep ocean. Areas within national jurisdiction include the land area, the territorial sea, and the exclusive economic zones, which stretch up to 200 nautical miles from the coast. Finally, we have the areas beyond national jurisdiction, also referred to as the high seas or international waters. Countries have full sovereignty within their terrestrial boundaries and in the territorial sea. This means that they can regulate the sources of pollution and the activities that lead to pollution in whichever way they want, subject to the international and regional agreements they've signed. Within the EEZ, a country's rights are somewhat reduced to the activities of exploring and exploiting the resources, such as mining and fishing. Beyond these areas of national jurisdiction, in the high seas, countries may conduct certain activities subject to the conditions specified in the Law of the Sea Convention, such as conducting scientific research and fishing activities. Vessels in this zone operate under the regulations of the country to which the vessel is registered, including the pollution regulations of that country. So from this, you can see that a country is given diminishing capacity to exercise and enforce its national laws the further out from land we go, which is also why the opposite is true for the duty to protect and conserve the oceans, which gets stronger in the convention the further out we go. Diving deeper into the Law of the Sea Convention, it's important to note that plastic pollution is not directly referred to in the convention. Remember, it's a framework agreement, so it wouldn't single out just one of the many pollutants, but plastics are inferred in a few articles. The convention provides a general overarching duty to protect the oceans. It also broadly requires prevention of pollution from all sources, preventing transboundary harm and protecting threatened species. Article 207 is the only regulation we have in any binding agreement at the global level that establishes a duty to prevent marine pollution from land-based sources. There are some at the regional level, which we'll touch on later. We've had some success on the sea-based sources of pollution through two agreements that give effect to Articles 210 and 211 of the Law of the Sea Convention. These require states to regulate dumping of waste generated on land and to regulate the discharge of operational waste generated on vessels while they are at sea. For the first of these two agreements, we can see that the London Dumping Convention applies to the intentional dumping directly into the ocean of plastic wastes that are generated on land. A permit must be obtained from the government before wastes can be dumped into the ocean, but any waste that contains plastic is banned from dumping in all jurisdictional zones. The second agreement that addresses sea-based sources is the Marpul Convention. The fifth annex to the convention deals with operational wastes that are generated on a vessel while it is at sea. It specifically prohibits the discharge of any wastes containing plastics in all jurisdictional zones and includes fishing gear. Vessels must report any losses, and if over 100 gross tonnage, they must keep a garbage management plan that can be inspected. In addition, if the vessel is over 400 gross tonnage, a garbage record book must be kept that logs the disposal of waste at port reception facilities. Still with sea-based activities, the UN Fish Stocks Agreement seeks to minimize catch by lost or abandoned fishing gear. But if it only applies to vessels harvesting particular fish stocks in the high seas and EEZs that are highly migratory. It doesn't specifically prohibit discharge of fishing gear and any other impacts this gear may have, such as destruction of habitat, which you've heard about earlier. But perhaps Marpul Annex 5, which we discussed in the previous slide, will close this gap. All the previous instruments highlighted are legally binding on countries that sign up to them. We also have the voluntary FAO Code of Conduct for Responsible Fisheries. It applies to all types of fishing in all zones and promotes, again, the minimization of catch by derelict fishing gear. It also encourages adoption of national regulations based on Marpul and promotes environmentally safe fishing practices, including reducing any impacts on associated or dependent species. So a little bit broader, but bearing in mind it is voluntary. Moving to the regional level, you can see here there are 18 regions defined under the Regional Seas Programme of the UN Environment Programme. These programs aim to preserve the marine and coastal areas within each of their geographic regions. Their legal frameworks vary considerably. It's a bit small to see here, but some regions have adopted binding conventions, and some of those have adopted protocols to these conventions that address land-based sources of marine pollution, including plastics. A majority of the regions now have adopted regional marine litter action plans, which are voluntary, except for the Mediterranean region, where the marine litter action plan is binding. So finally, this diagram provides a summary of all the relevant binding and voluntary instruments for plastic pollution that we assessed for the UNEA 3 report in 2017. You can see the geographic scope, which varies greatly, and combined, they don't cover the full life cycle of plastics. So I believe we have sufficient regulations for ocean-based activities, but they need greater enforcement, as we've heard tonight. Our big gap is an agreement for land-based sources, but international discussions have placed a lot of focus on this, and there's strong hope that at the intergovernmental negotiation committee will be established in February at the next year at the UN Environment Assembly meeting in Nairobi. Most of my current research is on this, and I focus more on what a new agreement could aim to achieve. So with that, I thank you, and I look forward to discussions.
Thank you so much, Karen. You gave us really clearly all the complexities that surround litter and pollution in the ocean environment. And thank you so much for doing that so clearly, but sharing with us all of those different aspects that are involved, the complexities and how they interlink. So we've only got a few moments now, and we've just got 3 questions or 2 questions that have come through. So maybe we can just address these. Firstly, thank you to all the panelists that have spoken today and your words of wisdom. We've got a question here from Mackenzie. Mackenzie says, what are going to be the source of pollutants and subsequent impacts of deep sea mining? Who would like to answer that? Have a volunteer?
Maybe Inês?
I'm here. Yeah. I pretty much believe that one of the release pollutants would be the metals that are trapped in minerals. When we explore the deep sea, you have to scramble the seafloor, and a lot of these minerals that are complex that become available to the seawater, and how they react in such different environments, more oxygenated and more cold, how can react to the chemistry of seawater and become another source of pollutant into deep sea fauna is a very good issue. Other sources are not direct with pollutants, but the impacts of the particles and the plumes that mining will discharge to the water column. So the mechanical and physical effect would also be a huge problem.
Great, thank you so much. We just got one other question, and it'll be interesting to hear from the panelists if we think there's an actual answer to this. And the question comes here, what is the most effective method of getting rid of microplastics from deep ocean hotspots? So maybe I will also rephrase that to say, do we think that there is actually any method of doing this? It'd be great to have people's just words. Does anyone think, firstly, that there's an effective method? And they're saying definitely no. Any of the other panelists think that there is any method that's likely?
Yeah, I tried to answer.
Thank you, Chari.
Well, the question is that where to find them? You know, we haven't even mapped the bottom of the ocean for looking at the bathymetry. You know, we're looking at large areas which are unmapped. So even if we actually, you know, that's the first one, as I can't remember who was it, Shamik said that, you know, we know more about the moon than the bottom of the ocean. So, you know, microplastics, the amount of sampling is so small. Right? It's not like looking at the ocean on the surface. But then the other one that, you know, a positive one to a certain extent is that most of the microplastics are buoyant. They would stay on the surface. No. So yes, we do find some fragments on the seabed in sediments, etc. But for a large amount, they would actually ultimately end up in the middle of the garbage patches or on beaches, basically.
Great. Thank you, Chari. Anyone else got any other comments on this? Yeah. So I think I'll just sort of add my words there. I agree, we've got multiple challenges, as Chari said. You know, we don't know what's in the ocean necessarily. We know that there are microplastics distributed from the surface to the depth, but we also know that that's where biology lives. And microplastics are often the same size as some of our smallest creatures. And we know from studies and experiments that have tried to remove microplastics from the environment, this has come at a cost to the biological life that's been in those areas as well. So it is a definite challenge. And I mean, I certainly would advocate for minimizing pollutant getting into the ocean rather than trying to clean it up, because that's far more effective in the long term. Well, I would just like to say thank you very much to everybody who's been part of this event today, to our participants who've been listening to us and been sending some great comments in the chat and in the questions, and of course to our wonderful speakers who have taken time to share with us their awesome research and their really important insights. So thank you so much, everyone. And I just want to say thank you so much for my personal thanks, but also thanks from Doce. And if anyone's got any further questions, they can find us via the Doce website. Thanks so much, everyone, and goodbye.